Thank you so much for having me. I'm really excited to talk to you about my PhD project, which is supported by, as Suresh said, supported by Australian Rotary Health, the Rotary Melbourne, and the Lawrence Laboratory. As you all know, mental illnesses are widespread, affecting 25% of the population, with 300 million people affected by depression, anxiety, and PTSD. And the ripple effects these conditions have on individuals and the people around them has motivated biomedical research into ways to treat these conditions. Biomedical researchers aim to develop drugs that can treat people living with mental illness. And before we can test these drugs in humans living with mental illness, we first need to test these drugs in animals, such as mice, living with conditions that mimic human mental illnesses. And now I'm going to go into an example of a human mental illness, which is alcohol use disorder, and how it can be simulated in mice for research. So let's take some mice living with withdrawals from alcohol dependence after a lot of alcohol exposure over time. Researchers, may treat them with an experimental drug and then ask the mouse whether, has this drug improved your withdrawals? But of course, mice can't talk, so we can't do this. So instead, what happens is we treat mice with an experimental drug, and we videotape mice running around an open field with and without a drug. And then researchers watch these videos and manually score the time spent doing withdrawal-associated behaviors like scratching and shaking. And then they compare the time spent scratching and shaking with and without a drug. And then from this data, you can conclude that whether the drug reduces scratching and shaking symptoms and recommend this drug for human trials. But there's a big bottleneck in this process, which is it's really time consuming to watch videos of mice and manually score behaviors. If you have like 40 hours of videos of mice, it's going to take you at least 40 hours to go through all those videos. And imagine what another member of the lab has to do. And now consider how many thousands of labs we have globally studying alcohol use disorder. You can just imagine the massive time loss there is globally. And if we could just take away this time loss, we would have so much more time for doing new experiments into the development of drugs for other mental health disorders. And that's what I want to do for my PhD project. I want to use AI to create a tool that can watch videos of mice and automatically classify the time spent performing behaviors. And this is not just important for alcohol use disorder research. This is equally valid for nicotine dependence research, if you see in the top left, opioid dependence research. And if you study different behaviors to scratching and shaking, you can all, such as grooming and huddling, you can also study mouse models of anxiety and depression. So this just shows that what I'm working on will hopefully speed up the preclinical research into almost all mental health research. Almost all mental health research. But I not only want to make a tool that can just score grooming and huddling or a small number of behaviors, I want it to classify as many behaviors as possible. So if you have a video of a mouse and you can really extract many, many different behaviors. And within each of these behaviors, you can get subtypes like scratching with the nose, scratching with the hind legs, scratching fast or slow, etc. And I want to make an AI tool that can extract all of this behavioral data at once. And this is important because let's just say you have a mouse model of alcohol use disorder, and you want to work out whether this drug reduces withdrawal-like symptoms like scratching and shaking. It might show that this drug reduces scratching and shaking, but what if it causes excessive grooming, which could be related to some other kind of adverse event, which we would only then discover if we did human trials? What if it causes excessive rearing, which you'd only discover later as some kind of hyperactivity, symptom in humans? So that's the importance of creating a tool that can classify all different types of mouse behavior simultaneously. And it's also really tedious to classify all these different types of behavior as a human. So in summary, I want to go from create an artificial intelligence tool that can go from a video of a mouse to a video of a mouse with all the behavioral classification data, so that this can speed up mental health research and provide detailed data about how drugs influence mouse behavior, which will then open a window into how these drugs may treat humans with mental health disorders. And I'm extremely grateful to Australian Rotary Health for allowing me to pursue my passion, which is this PhD project. Thank you.